Hi, welcome to a Physionic detailed study analysis. Today we're going to be going over 5 different studies, one of which being a meta-analysis, including more studies than the 5 that we'll be going over. And we're going to try to determine the effect that creatine supplementation has on our memory. But beyond that, we're also going to be going over some of the other neurophysiological effects of creatine. Now, this is a lengthy, detailed study analysis, so if you'd like to skip to the conclusions, please feel free. However, you will be missing out on considerable context. However, I've grouped all the highlights into 1 or 2 sentences at the end of the analysis for your convenience. For those nerds that would like to continue onwards, well, if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, hold my master's in exercise physiology. I've actually written a book on creatine, and this is what I do. I take studies or groupings of studies and try to come to some form of consensus, some form of understanding. That doesn't mean that you have to agree with my conclusion, but I'm going to present the data and then you can make your own mind up. Okay, with that out of the way, with that introduction out of the way, let's go ahead and jump into this. So the topics that I'm going to be covering for you are the mechanisms of action. So what are the proposed mechanisms that creatine has on our brain? Then we're actually going to look at the real world effects of creatine on our memory. Then I'm going to really intriguing difference, look at potential differences between men and women. And then we're going to look at an age effect, if being older or being younger, if there's some sort of an effect there. And then if you're part of my insider program, you'll also get access to the dose type, so the way of consumption, and some other really intriguing nuances when it comes to creatine supplementation. Then creatine on sleep deprivation and stressful situations, and some of the other neurological effects that could occur from creatine supplementation. If you're interested in having access to the full version, as well as a whole host of other benefits that are included in the Insiders program, along with an Insiders podcast, including other study breakdowns, research reviews, and a whole host of different things, live sessions with me, then certainly you're welcome to join the Physionic Insiders program. But if you're interested in just the main outcomes here, so looking at creatine's memory effect, then let's jump into this. So the mechanism of action. So let's get into the cells. Let's, as we do in Physionic, let's dive into the intricacies of the cell. So we've got our brain here, and let's say we were to look at a particular neuron, so just one brain cell, and then we were to zoom into that brain cell and we were to look at just the cell membrane. Here we've got our neural cell membrane, got the outside here, and we've got the inside here. So this is the cytosol, and this would be the extracellular fluid. Now, inside your cells, you have ATP. You have these molecules of adenosine triphosphate, and they are known as the currency of the cell. They are the energy currency of the cell. Now, there are other molecules as well, for example, like GTP, which are also energy currencies of the cell, but the main focus for a lot of biology is focused on ATP specifically because there's so many reactions, so many more reactions that occur with ATP. So, how do we generate ATP? Well, we can go through a normal process known as glycolysis, which is taking glucose molecules from outside the cell. They enter inside the cell, and then they go through the glycolysis pathway. So they get this glucose molecule gets acted on by a series of different molecules, enzymes, and those change the structure of glucose. But as they do that, they start to spit off and start producing ATP. Now, additionally, glycolysis can go into the mitochondrion through a process called aerobic glycolysis, which can ultimately lead to more ATP than just anaerobic glycolysis. So, but the point is that it can go through this metabolic pathway called glycolysis and can ultimately add to the pool of ATP that's available within the cell. Now, beyond that, however, there are other molecules that can enter the mitochondrion. So like astrocyte metabolites. Now, the reason why I'm pointing this out is there's a very special distinction between neurons and some of your other cells, the majority of your other cells. If you were to take a muscle cell, if you were to take a liver cell, for example, it would be able to take up fat molecules and then be able to put those in the mitochondrion and generate ATP. However, your neurons have a very limited to no capacity to do that. So what they do is they rely on other cells called astrocytes to take up those fats, to generate those metabolites, which would generally be made by the cell itself inside the mitochondrion or just outside the mitochondrion. And then because it's relying on these metabolites, these astrocytes have to kick those metabolites out. It's almost like you can think of it like a bird pre-chewing the food for its babies. The neuron is the baby, and the adult bird is the astrocyte. So the astrocyte is supplying the chewed up food. So the chewed up fatty acids in this situation, the chewed up fat molecules in this situation, and turning them into NADH or other energy producing metabolites, which ultimately get exported out of the astrocyte and then enter inside the neuron itself. And then those can then go into the mitochondrion and generate ATP. So it's a very symbiotic relationship between the two, although the neuron's a little bit more selfish than the astrocytes. The astrocytes are very giving. Okay, so what do we use ATP for inside our brain? Well, there are a whole host of different functions for our brain. However, one of them is in the exchange of ions across the cell membrane. So again, this is the outside of the cell, this is the inside of the cell. So ATP is involved in the sodium-potassium pump. So pumping sodium back out of the cell from the inside to the outside of the cell, and allowing potassium back into the cell. And we have different leak channels as well, but for the most part, when we're talking about ATP intensive processes that happen within our cell, this is one of the major ones. And this is the one that I'm going to highlight specifically for this conversation when we're talking about creatine. So the point is that you usually have a high gradient of sodium outside of the cell and a relatively larger gradient of potassium inside the cell. When you look at the absolute values, it's a different story, but as it stands, without getting too confusing, just know that there's more sodium outside the cell. There's more sodium outside the cell and more potassium inside the cell. So the cell has to continuously make sure that there's the right amounts of both on either side of this cell membrane. And it does that through this potassium pump. Now, or sodium potassium pump. And as such, it uses large quantities of ATP to facilitate that function. So to give you an example here, so we've got a neuron here and we've got a neuron here. And when they're in their quiet state, so they're not activated, so they are in their quiescent state, then you have the sodium that's outside. You've got a sodium channel here, and you've got a potassium channel here, and you've got potassium that's found inside of the cell. We've got our cell membrane, outside the cell, inside the cell. Now, for a signal to propagate and to move all the way down this first neuron and then to communicate that signal across neurons from one cell to the other cell, you have to have this mass exchange inside this neuron of these electrolytes of sodium and potassium. So suddenly sodium will rush into the inside of the cell. This changes the chemistry of the cell itself to the point where you have this reaction. And this is, I'm being very general here because that's not the point of this conversation. The point is that just all you need to know is that there's a reaction, an activation of the cell when sodium enters the cell. Okay, that's all you need to know. And what also tends to happen is the potassium is then effluxed out of the cell. So this whole process is called depolarization, repolarization, and hyperpolarization. Those words themselves, if you're in biology, you will recognize them. If you're not in biology, you may not recognize them. It doesn't matter. The point is this whole process, this exchange, this reversal from the quiescent state. Remember, the quiescent state was with the sodium outside, the potassium inside. This reversal where sodium is suddenly inside the cell and potassium is now outside of the cell in very relative basic terms. So there's a lot more specificity here. I just have to be clear. Then that leads from a quiescent state to an activated state. That activated state means that there's a signal that gets propagated all the way down the cell and then eventually gets carried over to this next cell and then activates that cell. So how does the cell then return back to its quiescent state? The way that it does that is by these sodium-potassium pumps and through these leak channels. So the sodium will then be kicked back out of the cell and the potassium will then be allowed back into the cell. And this is an energy-intensive process. And if this has to happen millions and millions and billions of times as each neuron is communicating with another neuron to essentially form memories and to form different neurophysiological functions for the brain, then this is an intensive area of energy use. Okay. So let's return back to this. Nothing has changed here. We've got ATP that's very involved in the sodium-potassium pump. Nick, how does this relate to creatine? Well, it relates to creatine because the creatine molecule can actually take up what's known as a phosphate. Remember, I called this the adenosine triphosphate, meaning that it is a molecule that is an adenosine molecule plus the addition of 3 phosphate molecules tagged onto it. Now, that is a fully charged ATP molecule, and that can then be used for cellular energy. So what happens when you use the ATP? Well, the ATP gets degraded or gets cleaved, gets changed from an ATP to an ADP. So adenosine triphosphate, 3 phosphates attached to an adenosine molecule, to adenosine diphosphate, so 2 phosphates. So you lose a phosphate. Guess what creatine does? Creatine can be phosphorylated by an enzyme called creatine kinase, which ultimately leads creatine molecules to now be turned to phosphocreatine or creatine phosphate, but usually people say phosphocreatine. So this phosphocreatine, guess what that does? Because it has a single phosphate on it, what do you think it can do to ADP molecules? It can re-energize them. You can offer, the creatine molecule can essentially offer its phosphate to the ATP and re-energize it back to an ATP. So therefore you get the recycling of energy, and that's the beauty of creatine, that you don't have to go through glycolysis to generate new ATP. You don't have to go through the mitochondrion to generate more ATP, which would require more substrate. You can just use creatine as a tertiary system to generate more energy. Now, if you have more energy, what do you think happens here? That means then you have more energy available to have this quick communication between these cells, between these neurons. And that ultimately leads to better overall neurophysiology function. Now, that is the canonical system, and that is only a simplistic, that's the major system, but there are other mechanisms by which creatine can also have these effects. I'm not going to be going into them here, but just as a few things, creatine can change gene expression within the cell, can lead to greater expression of particular proteins that would be pro-memory. It can also act as an antioxidant. It can also protect mitochondria by globbing onto mitochondria and making sure that they don't end up bursting. There's a number of different kind of less known mechanisms that creatine can have an effect on the brain. But as it stands, this is the primary one. So it's an energy-driven effect. Now, let's talk about creatine on memory. What are the actual real world effects? It's interesting to go into mechanisms. I always love going into mechanisms. I always make sure to have that for these study analyses so that you can understand why it's actually having an effect, if there's an effect. But it's also important to look at the data itself. So we're going to be relying a lot on this meta-analysis. So this meta-analysis, which I've labeled Study 166, did not have any funding. So there's no external funding of any sort. I don't even think there's an internal funding. It's just a group of researchers that are aggregating the data across a number of studies. In this situation, we're talking about 8 studies that were included in the meta-analysis. You can see their rubric here for how they ended up accepting certain studies. So they had exclusion and inclusion criteria, which are right here. So the inclusion criteria had to be in healthy adults, had to be creatine monohydrate supplementation. So we're not talking about creatine, some other forms of creatine, like creatine HCl is one example. The comparator had to be a placebo, so it couldn't be like omega-3s or something like that. It had to be a placebo. And they were looking for memory performance outcomes. And this was restricted to randomized controlled trials. So we're talking about actual cause and effect kind of studies. Now, the exclusion criteria would be obviously in many instances the opposite. So non-randomized controlled trials, cohort studies, etc., in vitro studies, so talking about cell studies, looking at other outcomes related to cognitive function. So maybe reaction time is another example, which we'll be getting into if you're part of the Insiders program. I'll be going into a little bit on reaction time and executive function and a number of other brain indices. But they end up excluding those in this analysis. Non-placebo control. So if it doesn't have a placebo in the study, then they exclude that. So if I can go back here real quick. So they're excluding certain studies because of that, ineligible outcomes, incompatible populations, maybe populations with comorbidities or dietary restrictions. Maybe it was just in vegan individuals. So that was part of their exclusion criteria, so they removed that. Okay, so what did they find? I should note here that these researchers had to correct their analysis. Initially, they had a particular analysis that included many, many more of different outcomes. However, one of the drawbacks of that was that it is statistically improper to have multiple measures of memory coming from the same sample. What do I mean by that? I mean that if you are going to do an analysis, a meta-analysis of this sort, you have to make sure that each one of these data points, each one of these studies has a unique sample, so a unique group of individuals that were studied. You can't report multiple measures of, let's say, memory. So let's say they do test A, test B, test C on one sample, one group of individuals, because that's bolstering the data without actually having unique individuals involved in the data collection. Hopefully that makes some sense. It's a little difficult to explain, but the way it's supposed to work is that you have one sample, you're allowed to take one measurement and use that one measurement in the meta-analysis. If you have one sample and you take multiple measurements, then you are artificially kind of inflating the results of the analysis. That's the best I can do on the fly for an explanation, so excuse me. So they ultimately at first indicated that there was a benefit of creatine supplementation on memory, just general average memory. However, when they got called out by other researchers for this significant statistical error, they ultimately redid the analysis, and that's what I'm showing you. So I'm ignoring the data where they did a lot of these repeat measures across multiple test criteria, or these multiple tests in general. So this is all the studies, and they took one test from each sample, and ultimately what did they find? Well, to read this, I'm not going to go into all the details of how to read this and whatnot, but these are all the studies listed out. Here's the mean under the creatine group and the mean under the placebo group. They take the difference between that, so standard mean difference, so SMD, and then they end up reporting it. The actual visual of all these numbers here is listed right here. So the green is the indication of the mean, so the actual average result that they ended up finding, and the lines on either end are the confidence intervals, so how certain are the results around this mean. And ultimately, this line down the middle is 0, indicating that there's no effect. If all the studies move more towards the left, then that means that there is a harmful effect of creatine on memory. And if the results move more towards the right, then that means that there's a benefit of creatine on memory. So that's because the placebo, in favor of the placebo, would move to the left, which would then mean a detriment to creatine. And if it moves to the right, then that's in favor of creatine, as we see here. So it seems like if you were to look at this, if you were to aggregate all this data together, and then they do that by putting that into this black diamond. This black diamond is taking all of the results of all these studies, all these numbers, and just aggregating it into one really easy way to just look at the average result across all the studies. And what you would think is that even though you have a few studies that maybe lean a little bit more to the left, you have one study here, this Rawson 2008 study, that indicates there was essentially no effect. Maybe the same could also be said of this study. The Merej-Filho study would also say that there's maybe no effect, maybe a slight detriment to creatine. But you also have a few other studies that indicate that are strongly in favor of creatine. So in aggregate, what does this tell us? Well, if we look at the diamond, it seems to move a little bit to the right in favor of creatine. However, if we actually don't trust our eyes and we use the statistical measures here, we indicate that actually there is no statistically significant effect. So they went from a position of, yes, there is a benefit of creatine, to technically now there's no benefit of creatine. And that's after they've removed all of those confounding measures that they did in their initial analysis. So with a p-value of 0.15, it's starting to maybe trend towards that direction. And maybe if they had more data, they might be able to get, which clearly they did, but obviously they had to remove it. So more unique data, I should say, then maybe they might see an effect. But as it stands, there does not seem to be an effect of creatine supplementation on the brain. Hey, so we're about to go into the aging effects and the effects or differences between men and women when it comes to creatine supplementation. However, if you're interested in actually going into dosing as well, the types of dosing, looking at the effects with sleep deprivation and other neurological effects of creatine, then I would strongly emphasize that you join the Physionic Insiders, where you'll get access to the full version of this video, as well as a whole host of other benefits that come with the Insiders, with live sessions with me, an Insider podcast that releases every month where I aggregate all the data for that month's investigations with applicable takeaways and a bunch more. So if you're interested, certainly feel free to join, and you can find that in the link down below. With that, let's get back to the video. So the next question we have to ask ourselves is there an age effect? So it's one thing to look at the overall effects and then say, okay, well, then I guess we can ho-hum, we can just go home. But in reality, analyses in science is a lot more sophisticated than that. And we can actually do what are known as subgroup analyses. So not only look at the overall effect, but start to break out studies and try to separate out studies based off of particular characteristics. In this situation, we're talking about age. So it's the exact same read. We're doing the exact same thing, but we've broken it up into two different groups. One group is younger adults and children, which they've listed all the studies there. And then they've got older individuals. They've got two studies here. So now we're breaking it up by age, and the age cutoff point is 65, if I remember correctly. So if we look at the younger adults and children, we see that all these studies are relatively in line with one another. And we can see that the diamond is smack dab in the middle, indicating that there's no benefit or harm of creatine supplementation. Now, if we look at the p-value, there's a strong indication that that is correct. So the interpretation, what we would see here, what I would see at least, I'm only speaking for myself, that would indicate that there's no effect of creatine supplementation in younger individuals. Now, on the other hand, for older individuals, what we see is that a strong shift towards the right. And remember, what does that mean? Well, that's in favor of creatine. So it seems like the overall effects diamond is moving towards the right. Now, if we check the statistics, what does it show? It shows a strong p-value of 0.004, indicating that's likely a real effect. So it would be nice if they had a few more studies in here, but as it stands, these two studies strongly say that for older individuals, those over the age of 65, that creatine supplementation is a benefit to memory. So think about that. What a remarkable discovery to go from, creatine has no effect, to now breaking it up by age, and now you're actually seeing an effect. And that's where those nuances are incredibly important. So for older individuals, there is an effect. Now, what about differences between men and women? So here what they did is we've got males and females together. Okay, then we've got the separation of just females, and then we have the separation of just males. Now, for males, we only have one study that's specifically only males, as opposed to samples that include maybe 15 men and 10 women, or 20 women and 11 men. So those would be the females and males. And females studies is just women. So, maybe 10 women and no men, obviously, or 20 women and no men. So because of that granularity, now we can actually try to aggregate the data and try to figure out, okay, well, if we separate the data based off of those groupings, do we see an effect just like we did with age? So in the combination of men and women, we see that there's no effect. So it slightly leans towards the right, but if we check the statistics, it's a p-value of 0.36. So there's no effect. There's a little bit of heterogeneity for anybody that's interested in statistics. And that's probably just driven by this McMorris 2007B test, or study, I should say. And it's still not very high, though. Now, for women, again, there's only 2 studies in this situation, but it seems like with these 2 studies, it's starting to trend towards maybe there is an effect. But unfortunately, the p-value indicates that there's no effect. So the proper conclusion, also with no heterogeneity, the proper conclusion is that there's no effect in women specifically. And then when we look at men only, of course, there's going to be zero heterogeneity because it's not applicable. There's only one study, so we can't compare between studies because there's only one study. You can't compare against itself. And for this, we see that the effect is there's no effect. Now, one question that might be crossing your mind is, well, okay, we're looking at men and women together, we're looking at women separately, and we're looking at men separately. Why do we suddenly see no effect when earlier we did see an effect with the age? Because obviously these have to be men and women, right? Well, the reason for that is because here they're separating out based off of age, and here they're not doing that separation. So they're mixing together all the young individuals with the older individuals all grouped together. So that ultimately dilutes the effect that we saw up here with the older individuals. That's the reason why. Okay, so the midpoint conclusion at this point is that creatine is a benefit to memory in older individuals, those who are 65 or older, but does not seem to be a benefit for younger individuals. There's likely a need for more studies, however, and probably no real difference between men and women, but we definitely need, we absolutely need more studies in that regard. If you're interested in joining the Insiders program, I'm also going to be covering dosing creatine appropriately for those older individuals. Also, some additional considerations. There's more to this story than I'm letting on, but as it stands specifically for memory, this is it. This is the story. But in terms of other measures, also what effects could creatine have as an anti-stress effect? And creatine on sleep deprivation effects, which are really fascinating studies. So if you're interested in accessing this, then certainly just jump on over to my Physionic Insiders program. You'll get access to this plus a whole host of other benefits as well. So it's not just this. Although, I still think this is good work, but still, regardless, you'll get a lot more than that, like my Insiders podcast, where I aggregate all the data for the month with applicable takeaways, and I also make sure to answer your questions as well if you're a Physionic Insider. But, no pressure. Hopefully you found this informative, and I'll hope to speak with you in the near future. Have a good one. Bye.